Is probably better to title it Rediscovering and Enhancing Old Tricks of an Old Dog. So DMS is a classic chemical probe, and we're sort of doing things that people did with DMS many, many years ago, but have, I guess, been forgotten or because they were difficult. I'll try to touch a little bit on that history. We're interested, I think, as everyone hopefully on this webinar is, in RNA structure, how RNAs fold, because that's really intimately linked to how they function. And so it's not just how RNAs fold, but also their dynamics that can be important. Just sort of in this diagram here, as we're thinking about an RNA, we've got potentially some secondary structures. Inside of a cell, there might be proteins that bind to some of these structures. We're going to have potentially tertiary structures. This might adopt, totally different secondary structures. We have, switching between different ensemble states. But then also within a given structure, there's going to be sort of smaller fluctuations like base pair opening and sort of 3D unfolding. And so if we really want to understand how RNAs work, I think, need to understand basically this entire landscape of RNA folding and its interactions with cellular partners. And so ideally, we would develop predictive models that allow us to just take sequence and get all of this information. But as we know, that those models are not currently where they need to be to fully answer this problem. And so that means that we use chemical probing experiments as a way to get at some of this information. And I think many of you are familiar with these experiments. These use shape reagents or DMS are sort of the two most commonly used chemical probes, and we can add these reagents to cells or RNAs and test tubes, and they will react selectively with usually single-stranded nucleotides, leaving behind these chemical adducts. And then we can read out these chemical modifications using sequencing-based approaches. Our preferred approach is this mutational profiling strategy where reverse transcriptase enzymes read through the modifications and introduce mutations in the cDNA. And so really the advent of sequencing and I think mutational profiling has allowed us to make these measurements, at very large scales and with significantly improved sensitivity. We can really measure small reactivity changes, which are going to become relevant later in the talk. These are, of course, very old methods, and that sort of goes back to my title. People have been doing DMS probing for many, many years, but mutational profiling has, I think, really transformed the field. After we can then sequence these data, look for where these mutations occur, and then compute a reactivity profile in the traditional way of analyzing these data, where highly reactive nucleotides often are shown in red, and we can infer our single-stranded, lowly reactive nucleotides shown here in black. We can infer our base paired. And we can use this data directly for analysis, or we can use it to do structure modeling. So the experiments are great, but they also have a lot of limitations. And, I think if you think about what we actually get from a chemical probing experiment, we're really just getting traditionally this one-dimensional measurement of reactivity. And really what we want to be able to do is actually measure this entire structural landscape. And so, this is a hard problem. And just to sort of emphasize some of the issues that, the data only provide a partial and indirect picture of structure. It's very challenging actually to identify from data like this whether a nucleotide is unreactive because it's in a secondary structure or because it's, participating in a tertiary structure or bound by a protein. And, that's potentially very different from a functional perspective, very different from an interest perspective if you're trying to think about designing something like a small molecule that's targeting an RNA. And then finally, it's challenging to quantitatively measure dynamics. So, for a variety of chemical probes have been developed over the years to try to suss out different aspects of RNA structure and address some of these limitations. This is a nice image from a review by David Mitchell and Phil Bevilacqua that was published a few years ago, looking at sort of traditional chemical probing approaches that show, some of all the various reagents that have been developed to probe different positions on RNA bases. I'll also say that, not captured in this picture is the diverse array of cross-linking approaches to measure three-dimensional structures. And so these all have, can be quite powerful and all have their place. But, from our perspective, it's sort of, undesirable to have to use a bunch of different reagents to get at different aspects of structure. We've got to do many different experiments. Each can, be challenging to do. Each is expensive. And so really, like our vision is, can we use one reagent to get basically all the information that we want? And the reagent that we've focused on is DMS. And DMS has a number of favorable characteristics. I'm not going to go into all of them, but I'll just sort of note two right here is that it's readily cell permeable. So shape reagents also can get into cells, but it's, really easy with DMS. But I think that the most important reason why we like DMS is that it allows us to get very high, if we want, high levels of modification on individual molecules. And that allows us to then measure these correlated modification events that tell us then a higher order information about structures like tertiary structures or direct base pairing interactions, and also things like ensembles. So we published a review with Kevin Weeks a few years ago, sort of talking about this approach. So we've been focused, or since I was a postdoc in Kevin's lab, I've been interested in trying to improve DMS and specifically address some of the liabilities that DMS has. And that's classically that it only reports on A and C nucleotides, that it's unable to distinguish tertiary from secondary structure. That's common to many chemical probes, and that it can't measure through space interactions. So, over the years, we've developed a variety of ways to try to address these problems, and I'll talk on about each of these to varying extents through this talk. Okay, so before I sort of get started talking about talk about the new stuff, I'll sort of rewind to work that I started as a postdoc in Kevin's lab, and that was trying to address this issue of DMS only being able to measure A's and C's. And so what we realized is that if we were able to do the chemical probing experiments under moderately basic conditions, actually at pH 8, which is the same pH that shape experiments are typically done at, that would enable transient deprotonation of G and U nucleotides. And that would then allow DMS to modify G's and U's at low rates. And so we did that experiment, and what we found is that if you look at sort of the modification rates that we're measuring at single-stranded versus paired nucleotides, we see, I guess at A's and C's, very nice separation here, where there's much higher modification rates at single-stranded A's and C's compared to known paired nucleotides. But we're also able to achieve sort of similar performance at U's and G's. The modification rates are much lower, which is as expected, but we're able to now make measurements of structure, base pairing status, at all four nucleotides. And so this is quantifying here the performance at discriminating paired versus single-stranded at A's and C's. And you can see that we're doing quite well here with an area under the ROC curve of around 0.85 or zero point 0.9. U's is actually just as good as A's and C's. But guanosine was so-so. Some RNAs it does okay, but others not so much. And so when I started my lab, I wanted to sort of revisit this sort of experiment here and try to optimize it. And so we did a systematic evaluation of the various RT protocols that were published at the time. And I'm not going to go through the data, but you can sort of read about it here at in this paper that was work led by David Mitchell. What we found was that marathon RT, which was a new reverse transcriptase that was recently discovered at the time by Anna Pyle, was the best for us for measuring these reactivities at all four bases. So some of the advantages are that we found that it's very processive, that was published. For us, it had low background error rates, fewest indels, and overall just greatest predictive power at sort of discriminating paired versus single-stranded nucleotides. And so that sort of comes, I'll remind you again of my conflict of interest that around the time when this paper came out, Anna started a company that is commercializing Marathon, and she asked me to be on the advisory board. So I definitely be aware of that, I guess, if you want to interpret things as skeptically. Okay, but one of the interesting observations that we found when we were looking at the marathon data is that when we specifically at G's, we observed that there was a real difference in the information that's encoded based on what mutation we observe. And so normally when you do a chemical probing experiment, we consider any type of mutation that we observe at a position to be equivalent. That is just a sign of that there was a modification there and that the reverse transcriptase enzyme inserted a random nucleotide. But what we observed is that that is not the case for marathon, that you can actually see here that looking at the ability to discriminate paired versus single-stranded nucleotides, these C and T substitutions actually have a lot of information quite specific to single-stranded nucleotides, whereas actually the dominant set of mutations that we observe are these G to A substitutions, which have no information in them at all about base pairing. And so what David did is he said, well, let's just focus on this, the stuff that has information, these C and T substitutions. And so when we do that now, we can actually get very good, we can recover quite specific information about base pairing that actually compares to, and in many cases outperforms what one gets from a shape reagent. So just sort of showing what that data looks like for a representative RNA. This is RMRP, non-coding RNA that we probed David probed in HEC293 cells. And you can see here this four-base DMS-MaP data that we're getting is, and I guess I should say the base paired nucleotides that we expect to be paired are shown here in gray. And you can see that there's just a really striking correspondence between the four-base DMS data compared to even what you're observing with shape reagents, and also just sort of comparing this to what you would normally get from a DMS experiment where you're excluding G and U information, many, over half of the molecule you don't get data for. So we think that, we're really advocates for this experiment. I think that you get a lot of value from doing it. The one thing I will note is that if you're trying to do this experiment, buffer conditions matter a lot. DMS decomposes in water into sulfuric acid. And so that means that you're going to have a precipitous drop in the pH of the reaction pretty quickly. And so if you don't have a sufficient buffer in your reaction, the pH is going to drop. That's going to significantly impact your ability to measure these mutations at U's and G's. So just be aware of that. Okay, so just wrapping up this part that we think that DMS can be used to probe all four bases well, and others have also found that and shown that. So it's not just our group. So if you're interested in doing this experiment, I think you should do it. Yeah, we think it enables best-in-class structure modeling accuracy, but there are some caveats. You do need to sort of tweak the conditions and be careful in how you're doing the experiment. So it is a little bit trickier than doing your standard probing experiment just at getting data at A's and C's. All right, so changing topics a little bit, what we uh, were really struck by, actually even more so when we looked at this marathon data, is not this C to T substitution pattern where that was giving us information about base pairing, but we wondered what is going on with this sort of surplus of G to A substitutions that had no apparent information about whether or not a base was paired versus single-stranded. And so we hypothesized that that was actually because DMS, what we were detecting at low levels was DMS modifications at the N7 position. And so it's widely known back in ancient times in 1960, but then going on into the early or late 70s that DMS is actually the position that it most likes to modify in RNA is the N7 position. And this is, utilized by Walter Gilbert and others to measure or to sequence RNA, but also to measure RNA, our tertiary structures. But the problem is that measuring these N7 modifications is usually hard because the modification is at the N7. The reverse transcriptase enzyme doesn't care about it traditionally. And so we thought, potentially that we'd stumbled into a way to measure these N7 modifications, and specifically that N7 modifications were being read as A, whereas these N1 methylguanine modifications were being read out as U and C's. And to test that, what we did is looked at modifications, natural modifications in the ribosomal RNAs. And you can see that that is recapitulated quite nicely, where these N7, natural N7 modifications are read out almost exclusively as G to A substitutions by, this is not DMS modified, we're just doing the same MaP reverse transcription across these modifications. But the caveat is that we're reading these out at very low efficiencies, right? These are modifications that we know are installed with basically 100% stoichiometry. So G to A does seem to be unique to M7 modifications. Conversely, if we look at an M1 methylguanine, what we see is that this is now read out with very high efficiency, almost exclusively as G to C or G to T substitutions. And so this is consistent with this idea that these two different chemical modifications that DMS gives rise to give rise to distinct mutational signatures. So what the question that we were interested in is, can we develop an easy way to map these N7 modifications? And then really, if we can do that, what information do these modifications provide? So I'll, say again that this is mapping N7 modifications is something that has, been done for, many, many years, but it's required laborious, biochemical protocols to access that information. And so we usually, or the field has usually ignored this potential modification, and the information that it might provide is not all that well understood. Okay, so this is work that was led by a postdoc in the lab, Irfana, and a technician, Thomas, and we put it on bioRxiv last year. Hopefully, it'll be published pretty soon. So you guys can read. There's a lot of information that I am not going to cover in this talk. So the first thing that we wanted to do is improve the ability of our mapping protocol to actually detect these N7 modifications. And what we reasoned was that the reason that these N7 modifications were being detected as A modification or A mutations is that they were inside the enzyme active site. They were being decoded as Ts. And we sort of reading the literature, there's support for this that basically an N7 modification is going to stabilize this enolate form of the guanine, which would then be decoded via the sort of the classical, standard base pairing interactions as a T. Conversely, we suspected that these N1 modifications are probably being read out as T and Cs through some type of syn base pairing interaction. So this is speculative, but what we were able to do is based off of sort of this insight that we could optimize the reverse transcription conditions, basically increasing the pH as well as finding the right manganese concentration that will promote this type of mispairing within active sites. And so that takes us now basically a five-fold enhancement of our ability to read out these N7 modifications. Normally, our readout is quite low, which makes it challenging if you're trying to really measure these in subsaturating or substoichiometric DMS modification conditions. But with this new optimized RT, we can get we think roughly 20 to 25% readout efficiency. So what that now means is that when we do from a single DMS probing experiment, utilizing these different mutational signatures, we can get two channels of information. So at all four bases, we can get information about what's going on at the Watson-Crick-Franklin face via this N1 position. And then via these G to A substitutions at Gs, we can get information about the N7 position, what's going on at the Hoogsteen face. So just to give you an idea of what this data look like on a nice representative RNA, this is a TPP riboswitch, classic model in the field. And so again, in a single experiment, what we are getting is these information at all four bases about N1, N3 reactivity, and you can see here plotted on the secondary structure that these correspond quite nicely with the known secondary structure of the RNA. But then we also get these measurements of N7 reactivity. And so here what I'm coloring as yellow are positions that are highly N7 reactive. And in purple are positions that we're seeing very little reactivity. And what I want to emphasize is that there's, you can see here that there's really not a lot of the correspondence or the N7 reactivities are poorly correlated with what's going on at the N1 position, supporting that these are measuring orthogonal positions or aspects of structure. So specifically, if we look at these Gs here, they are unreactive at the N1 position because they're base paired, but at the N7, they are quite highly reactive. And if we look at these now on the second, the three-dimensional structure, you can see that these are highly are solvent exposed, whereas these unreactive positions shown here in blue and purple tend to be solvent protected by the tertiary structure. So we extended this experiment to a broader or larger RNA to get more data. In this case, this is data that we collected on the E. coli ribosome in cells. And again, what you can see looking here at this N7 mutation rate, that there's significant variation in this N7 mutation rate here. Some positions are very reactive, but many are effectively completely unreactive. So we put a lot of effort into trying to understand and interpret these data. And the approach that we came up with is to effectively normalize these N7 reactivities, where we reasoned that what we really cared about are these lowly reactive positions. Those are positions that are indicative of something more interesting than just secondary structure. And I'm not going to go into the details. You can read about it if you want. But effectively, what we're doing is transforming this sort of raw N7 data into a normalized protection score, where the lowly reactive positions now are going to be highly we're highlighting these highly protected or significantly protected positions. And so I'm sort of going to be showing this data now using this scheme throughout the rest of the talk, where things that are very significantly protected, I'm showing here in purple, and you can see these coincide with basically the most lowly reactive positions in this segment of the 23S. And then things that are sort of moderately or still significant, but less extreme, showing here in pink. And just to sort of begin to draw you to think about what these data mean, is if we look at now this segment of RNA, you can see that there are some protections here or this segment of the ribosome has this region here that's highly protected, but then it's sort of intervening regions that are not protected. And if you actually look at the ribosome crystal structure here, you can see that this region that is unprotected, this helix 18, is this solvent exposed helix, whereas these regions of protection at this three-way junction here, helix 21 and helix 22, these are basically buried within the interior of the ribosome. So immediately sort of consistent with this idea that these N7 protections are providing a measure of three-dimensional solvent accessibility. And indeed, if we quantify this over the entire ribosome, we do quite well as at predicting or identifying solvent exposed, or I guess, sorry, solvent protected nucleotides. And I'll also just note that these N7 reactivities are largely distinct from N1 reactivities, that they're poorly correlated, consistent again with this idea that we're measuring distinct aspects of structure with this experiment. Okay, so we went ahead and extended it to a number of different RNAs, both in cells and in vitro. And what we find is that overall, we do very well at discriminating discriminating things that are solvent protected. So when we see a protection, it's very specific to there actually being some type of higher order interaction that's giving rise to a protection. Where sensitivity is also pretty good, although not as good as our positive predictive value. And so just a few examples of what these of the atomic level details of what gives rise to these protections. Here you can see a very strongly protected N7 position that is from the ribosomal RNA, direct hydrogen bonding with a ribosomal protein, We also observe protections from other types of complex internal loop motifs. So here there are two Gs that are N7 protected in the U1 SNRP that appear to be because of altered stacking interactions here. Okay. Just as a sort of final example of what this can do, or the specificity of this data, we got data on the tmRNA, which is this interesting non-coding RNA present in bacteria. And one of the things that why we like one of the reasons we like this RNA is because there's a number of pseudoknots. And you can see if we look at now in the N1 and N3 reactivities across the top, that we're able to discriminate what's base paired versus single-stranded quite well, but it'd be quite challenging to pull out from this data where are the pseudoknots. Whereas now with this N7 protection data that we get in the same experiment, we can immediately distinguish where these complex structures are. That this region here where there is no higher order structure, there's no protections, whereas each one of these pseudoknots is giving rise to these very strong protections, consistent with there being these complex folds that are protecting the N7 position from reactivity. Okay, just not really going to be talking about proteins here, but for the rest of the talk, but I'll just note that if we remove the proteins, this is a protein binding site here, this goes away. And so one of the applications of this experiment that we're quite excited about is to use it to measure protein binding sites in cells, but also as just a further control. If we now totally unfold the RNA, all of those protections go away. So, sort of two applications that we think this experiment has that we're excited about is using these experiments to discover new types of complex structures that normally are challenging to distinguish. So, sort of if I've got a random RNA here, the goal would be, can I figure out that there's a complex structure here, but sort of not maybe on the 5' end, and that would enable a variety of functional analyses and also probably have value as you're thinking about, okay, which parts of the molecule might I want to prioritize for doing small molecule screening or things of that nature. So the problem that we chose to focus on as sort of a nice model system was looking at the E. coli ribosomal RNAs under what we call cell-free conditions. So trying to understand what do these molecules look like in the absence of proteins. So work from Sarah Woodson, Draper, and Jamie Williamson, and others have shown that portions of the ribosomal RNAs can fold independently of proteins, and that there seems to be, potentially a correspondence between which regions of the ribosome are folding independently and which where ribosomal proteins, the earliest ribosomal protein binding events occur. But sort of characterizing this on a sort of molecular wide base or across the entire ribosome has been very challenging, because measuring these types of transient or semi-stable tertiary structures in the context of thousands of nucleotides, it's just not something that current methods are set up to do. So we did this experiment. We probed E. coli ribosome in the presence of 200 millimolar potassium, 5 millimolar magnesium. These are conditions where we would expect potentially, if there are going to be any independent tertiary structures, where they would form. And indeed, what we see is that there sort of throughout the molecule, we see these clusters of N7 protections. And in analysis, I'm not going to show. These tend to be very they're statistically significantly clustered in both secondary structure space, but also sort of in three-dimensional structure space if we project those back onto the known three-dimensional structure of the ribosome. So this is in the 16S ribosomal RNA. Also in the 23S ribosomal RNA, we see similar types of clustered protections. Again, consistent with this idea that certain parts of the ribosome are able to fold independently of proteins. And so then what was really interesting is then if we map these back onto the ribosome and compare those to the known binding sites of these earliest primary ribosome binding sites, you can see that there's almost a one-to-one correspondence between where these primary proteins are binding on the RNA and where there is sort of an independently stable tertiary structure. And so very much consistent with the idea that the ribosomal RNA is effectively encoding this hierarchical assembly of the proteins via these sort of tertiary structures that sort of are seeding that process. And just sort of zooming in on one of them, this is sort of if we look at the structure, from now the folded assembled ribosome and look at where these protections are on it, you can see that they map quite nicely, really consistent with this folding, or this looks basically like an independently stable tertiary structure to me. And then if you look at the binding interface for this early protein binding protein, L24, you can see that it is, seems to be recognizing that interface quite nicely. Okay, so sort of the second question that I'll touch on here is, can we use N7 data to guide 3D structure modeling? So this is something that we definitely think is a possibility that we're excited about. This is something that reviewers wanted us to do. It's ideally, I guess, we should be able to, one could envision using these N7 reactivities as sort of a solvent accessibility restraint to guide something like a diffusion model or another type of algorithm. And so we're really interested in that, beginning to think about that and work on that. But that's a pretty significant project, we think. So as sort of a different way at sort of getting at this question, what we did is we used these N7 reactivity data to measure what we call rings, which are correlated modification events in single molecules. And that basically will report on sets of nucleotides that become coordinately reactive or unreactive. We don't fully know the model, but I think a good framework for thinking about is that a tertiary structure like this might transiently open and close, and that's going to give rise to these correlated modification events that we can then measure. And so this was, the ring analysis was first applied over 10 years ago, looking at N1, N3 rings by Kevin's lab. And they showed that, they do a pretty nice job of recapitulating 3D structure. And so now what we've done is extended that also to N7 rings. And so what we see is that a number of correlated modification events that involve directly N7 so two N7 positions that become coordinately reactive or unreactive. We also see correlations between N7 modifications and N1 modifications. So sort of drawing these out, we call these ring networks. We can compare and contrast between the N1 N3 rings and the N7 rings, that there's many more N1 rings overall, but the N7 rings are a bit more specific to tertiary structures in particular. So we think that there's value added there. You can project these onto the three-dimensional structure, and you can see that these in general do identify relatively short distances. So what we did is combine these, so take knowledge of the known secondary structure as well as these rings that we're measuring, implement them as distance restraints, and then feed that into far, generate models and basically evaluate how well do these restraints improve structure accuracy. And in general, what we find is that uh, including this additional N7 data leads to an enhancement in sampling near native structures. We're still, pretty uh, doing worse for sure than the state of the art of AlphaFold. But this is just meant to be a proof of principle that, we do think that there's information encoded about the three-dimensional structure in this reactivity data that we can use moving forward to improve structure modeling. Okay, so summarize this that this new multi-site DMS-MaP experiment enables easy. So I'll emphasize that it's just a tweak to your RT conditions, allows you to now get this entire new dimension of structural information about RNA. These N7 protections that we're able to measure report on solvent protection due to diverse higher order structures. And so we think that this is going to improve discovery of complex structures and novel RNAs. And we think also has a lot of potential moving forward for improving 3D structure modeling. So happy to, if folks, we have our own ideas on how to do this, but this is a great community. So I'm very eager to explore all the possibilities for this data. Okay, so I think as I anticipated, I've gone sort of slow. So I'm gonna maybe touch on this last story, but then leave time for questions. I'll just, as a quick intro, I've talked mostly about um, static structures throughout this talk, but as I noted in the beginning, we're also very interested in dynamics. Uh, and we think that those are very important, or we know they're very important for function as well. And so we and others, I'll just note, have developed strategies to directly deconvolve secondary structure ensembles, so large scale secondary structure changes in RNA using single molecule probing strategies. I'm not going to talk about those today, but there's plenty of papers that are out there at this point describing this work. I guess I'm showing this image here. I will note that this deconvolution strategy does work with the N7 data as well. So we can measure these N7 specific protections in different states. But so what I'll spend the last few minutes on is talking actually about a different problem, and that's sort of measuring these more local, smaller scale fluctuations that RNAs undergo. And specifically that, if we think about a chemical reactivity, there's actually a fair amount of information that's encoded about thermodynamics. Particularly if you think about, let's say, a moderately reactive nucleotide, that this is going to implicitly, we know it depends on the probability that the structure of this nucleotide is sort of going to be single-stranded or it's going to be, reactive, as well as then some sort of unknown normalization factor. And the fact that reactivities encode thermodynamic information is, again, very sort of old knowledge. This is, I think, a really beautiful figure from one of the early shape probing experiments by Kevin's lab, where they're reading this out by gels. But you can see if you look at the shape reactivity of a number of these different positions in this case it's tRNA, you can see that it follows as a function of temperature. It follows this really nice folding a melting curve exactly as one would expect. So, again, this is, sort of pretty direct evidence that these reactivities do encode information about thermodynamics. And just in the past few years, there's been a number of papers from Joe Yesselman's group, also from Alicia Jones, and most recently from Lauren Hagler that have also shown that this, holds true in a variety of systems. And so, what we have been working on, actually in work that is led by a really amazing graduate student at Northwestern, Edric Choi, and Julius Lux out of Northwestern. This is a preprint that we just released I guess, a week or two ago, is trying to further extend and formalize this observation to try to directly extract thermodynamics without any type of external reference state. And the idea here is that the reactivity of a given base is going to be dictated basically by an equilibrium between an unreactive and a reactive state, as well as now the simultaneous degradation of DMS with water. Uh, so we can build a kinetic thermodynamic model of the reactivity process that basically is now where we can say that, okay, the reactivity of a nucleotide as a function of time is going to depend on these rate constants, the rate of DMS modification at sort of a single-stranded nucleotide, the degradation rates, as well as now this equilibrium constant between this unreactive and reactive state. And this is this thermodynamic information that we're really interested in. And so this is, I forgot to mention, basically directly inspired by the frameworks that are used to interpret hydrogen deuterium exchange experiments in protein biochemistry. So what Edric did to first test this model is he did a time course DMS probing experiment where basically, start a DMS probing experiment and then quench at different times and then measure the modification rates. And you get these nice curves here that fit beautifully to this reactivity model here. And what you can see is that the rates that we're measuring we're sort of getting two, or he gets two measurements. This K degradation rate, which is the rate of DMS degradation, and you can see that it's consistent basically across all the nucleotides, as well as now this K observed, which is includes this equilibrium constant that we care about, that there's orders of magnitude variation in this K observed rate here that seems to, as one would expect, uh, coincide with the known structural context of this RNA. So specifically at A18, this is a single-stranded nucleotide. You can see that we've got a very high rate constant, whereas the C25 right here in orange is base paired and has a very low rate. Okay, I'm sort of going to fly through this. I apologize, Edric, if you're on the call, but hopefully you have an opportunity to talk about it yourself soon. Um, so Edric did a huge amount of work to try to validate that the rates that we're measuring from these time course DMS probing experiments are physically accurate. And so just one of the experiments that he did is that he uh, obtained sort of a measurement of this K-add, or what's the rate of DMS modification of an unstructured nucleotide from using our prime experiments. And then he's independently measured it by NMR, by basically adding DMS to single nucleotides in an NMR tube and then directly measuring that rate of chemistry. And what he sees is that there's very good agreement basically between the NMR measurements and the prime measurements. And so this is just one piece of the validation that he did that we think or support that these are physically accurate rates. So with that in mind, then what he can do is he can transform now the data. And again, I'm really butchering this because I'm flying through it, but we can take now if our knowledge of this K-add rate, knowledge of this K-deg rate, which we can directly extract from the experiments to now relate the reactivity of a nucleotide to this inherent equilibrium constant, and then from that turn that into a delta G. That is basically the delta G of a base pair opening or closing in this simplified construct here, which is this 4U thermometer. And you can see that the delta G values that we get here, both for a wild type and then a mutant where we've stabilized this, make physical sense that they are between 2 to 3 kcals per mole. You can see that in this sort of wild type where we've got a mismatch here, that there is sort of local instability of these base pairs, whereas now if we make a substitution of this to a C, we see that this entire lower stem here sort of zips up, increases in energy. What's really exciting is if we correlate these measurements with independent NMR measurements of base pairing stability, we see really strong correlation. So, R squared of above zero point 0.8. So this is really consistent with these energies that we're measuring from a chemical probing experiment, reflecting the true physical energy of that base pair opening. Although, one of the interesting observations that we found is that these energies are about half of what are observed by NMR. And that is sort of interesting, suggests so it could suggest that our model is wrong. But we actually think our model is right. And as sort of inconsistent with that, there is sort of a different, where we are consistent with a different experimental methodology, the tritium exchange, sort of ancient, some of the first measurements of base pairing opening that were ever made suggested that the energy of base pair opening is 2 to 4 kcals per mole. And sort of this discrepancy between NMR values, which suggest it's more, akin to 6 to 10 kcals per mole versus these tritium exchanges has been a long-standing controversy in the field. And so it's sort of interesting now that our data are more consistent with this lower estimate. All right, in the interest of time, I'm just going to skip over this really interesting stuff, but just to say that we've been able to, Edric was able to extend this to now really complicated RNAs, or I guess P4, P6 is maybe not that complicated, but certainly a lot more complicated than sort of what NMR has been restricted to, which is, these simple hairpin constructs. And so he gets really interesting information now resolving energetic features at base pair resolution of P4, P6. And we can see, I guess I'll just sort of cut to this one of the interesting things that he sees is that there's appears to be long range coupling in the energies between nucleotides throughout P4, P6, but in also other molecules that we can now access with this prime framework. And so overall, there's still a lot of work to be done, but we're really excited about the potential of this prime framework to provide thermodynamic understanding of RNA, moving beyond sort of the heuristic interpretation frameworks. So I will thank the people who did the work, and particularly Irfana and Thomas, and then Edric and Julius. And with that, happy to take any questions.
All right, thank you very much for that wonderful seminar. For questions, raise your hand or put it in the chat. And then I'll try to condense questions together where possible in the chat so we can have more. I'll start with one from Shrikant in the chat. Is it possible to obtain both secondary structure base pairing information of DNA RNA hybrids, so moving to DNA, and its tertiary structure, so protein DNA RNA hybrids, using these DMS-MaP chemical probing experiments? So can you do it with DNA?
Yeah. I mean, yes, I think in principle, I mean, if you're getting the information on the DNA side is going to be challenging, but you can definitely there's nothing preventing you from measuring the RNA that's hybridized to a DNA, and so as long as you're okay with just getting sort of things from the RNA perspective, there should be no problems extending it to RNA DNA hybrids.
All right. So the next question from Kevin Weeks is, what are your thoughts on the ability of this N7 mapping to detect tertiary structure on a broader scale, like transcriptome level scale?
Yeah, thanks, Kevin. I mean, we're really we're trying to do that experiment right now. So yeah, I think that there's some going to be some challenges or it's going to require us to I think refine the analysis probably a little bit to truly reduce the false discovery rate to a useful extent. But we're definitely optimistic about that and working on that experiment.
And I think from the prediction perspective, having those kind of larger data sets are very useful for both training prediction and assessing how well people
Yep,
are doing predicting structures.
yep,
Um,
yep,
Okay, let me summarize the last two quickly. So from Horatio Guzman, he's asking, you show in cell secondary structure of N7 to track RNA protein interactions. Could you comment on how the cell-free conditions affect specifically tmRNA
yep.
secondary structure? And then from Simone Poblete, um, how does the 4-base DMS compare to CMCT for G as in use? So those will be the
Yeah,
final
so
questions.
the tmRNA, we our data support that these cell-free conditions have effectively no impact on the secondary structure. So removing the protein, that there's we only see like two changes in reactivity across the entire molecule, and that's exactly at that protein binding site. So there's not really, yeah, not affecting that folding. In terms of how it compares with CMTC for GNU, you know, I'll be honest, like I've not done like a really detailed, comparison. But I think it's certainly comparable. Is my anticipation. For us, the key we're not. I would not be surprised if CMTC has some advantages in certain contexts over DMS, and there's other reagents also for probing GNU. But to us, the real advantage and what we're trying to get out of this is that we get that information in a single experiment. So that's that's sort of our goal, and I'm not necessarily trying to say we're superior to CMTC at all times, just that it's easier to get the experimental data with our approach.